You can start. Yeah.
Okay. A very good evening to everyone. Myself, Farha Anjum. I'm working as a research associate at Department of Biotechnology, IIT Hyderabad. And I welcome you all in today's lecture on advanced fluorescence microscopy and image processing. Today's lecture is mainly a practice session on image analysis means whatever you have learned previous about image analysis by using image now you are going to implement all those methods in different types of exercises. Previously you have learned about the image software and loading of images, how you can load different type of images in this software. Also, you learn processing of 4D images, montage creation, manual time stamping, object counting, and how you can measure a distance in between the two objects. Apart from this, you have also learned how you can segment different complicated images by using ImageJ, and you have learned to use different types of filtering so that you can improve the quality of your images and you can reduce the background noise of your images. And further, you learn image macros. If you have huge images and several images, how you can automate different type of methods by implementing these image macros. And you have learned intensity measurements, co-localization so far. So here we are going to do this exercise. So before going to this, I want you to open your images software and the sample folders of images which we have provided you in Google Drive. So I'll give 1 to 2 minutes so that you can open this software and images. Okay, so for this exercise, what you need to do, you need to open one image which is present in your images software with the name confocal series. So for this image, what you need to do, you need to create a maximum intensity projection image, and then you have to change the color of different channels, and then you have to put scale bar. Before that, you need to do set scaling, and your final image will be look like this. Okay, so I'll start with opening of this confocal series image, and you can follow all these steps. You can do with me. This one is confocal series image. Okay, here you can see this one is of two channel image and it has 25 Z stacks. Okay, so first what you need to do, you need to create maximum intensity projection image. For this, what you need to do, you need to go to image, stacks and Z project. Okay, from here you have to select fifth to 20th stack, and here you will select maximum intensity, then click OK. Okay, so here this one is your maximum intensity projected image. Here if you want you can change its contrast by going image, adjust and brightness and contrast, but with this image this is fine, we can go with this. Next step is you need to change the color of these different channels. So for this, we'll go to LUT. For first channel, we'll change it with magenta. And for second channel, we'll click to the second channel. And then we'll go to LUT and we'll change it to the yellow color. Okay, so we have changed its color. Next step is you need to put a scale bar of 5 micrometer in the lower right corner. But before going to put this scale bar, what you need to do, you need to set scale. For this, you need to go to Analyze, Set Scale, and here you have to give these options. So distance in pixels should be 15, known distance should be 1, and aspect ratio will be 1, and unit of length micron. Okay, then I'll click OK. So next is you need to put scale bar of 5 micrometer. For this, you need to go again, Analyze, Tools, Scale bar. Here, we'll provide 5 micrometer of scale bar, and this value you can change if you want, but here I'll put height in micrometer 5, thickness in pixel 4, font size 14, and color of scale bar I'll provide white. Then okay, so now you can see this image is almost similar to this result image. Next, you need to save this as a .png with the name maximum confocal series. Okay, so I'll go to File, Save as PNG, and I have created this folder. I'll save this here. Okay, I'll wait for 1 minute so that you can complete all these steps. Meanwhile, I'll close this. Okay, so next exercise is creation of montage. So in previous lectures on this image processing, you have learned how you can create montage with different channels of images. So by implementing all those steps, we'll create this. So for this, what you need to do, you need to open this Neuron 5 channel image from this images software only. We'll go to File, Open Samples, and here you will find this Neuron 5 channel image. Okay. So in this exercise, you need to create a montage of 5 columns and 1 row. Before going to this, we'll change its brightness and contrast so that it can look beautiful. For this, you need to go to Image, Adjust, Brightness and Contrast. So here I can change this for first channel, then I'll go to second channel, I'll increase its minimum and maximum value, and then to the third, the blue one, and then to the fourth channel, and this one is TD image. So we have changed all the brightness and contrast of all these channels. Next, what you need to do, put a scale bar of 10 micros. So before going to put this scale bar, we need to set scale of this image. For this, what we need to do, we need to go to analyze, set scale. Okay. And then we'll go to analyze, tools, scale bar. So, okay, first we'll create montage, then we'll put scale bar. Okay, to create a montage, we need to go to image, stacks, and make montage. So here we have to create this montage for 5 column. Here we will give 5 and rows 1. Then we click OK. Here you can see this montage has created, but there is no line in between these images. Okay, so I'll repeat this. Image, stacks, make montage 5. So here we will give border width of 2. Okay, then now it's perfect. And next we need to put scale bar of 10 micrometer. For this we'll go to analyze, tools, and scale bar. Here we'll give scale bar of 10 micrometer, and then I'll click OK. Here you can see this is similar to this result. Okay, next we'll save this montage as a .png file. For this, we need to go to File, Save As, and PNG into this folder. Okay, here I'll wait for 1 to 2 minutes so that you can follow all these steps. Okay, I hope you all are with me. So here I'll close this and I'll go to next exercise. So this one is here also we need to open one image, but this you need to open from the sample folder which we have provided in Google Drive. Um, this image will be with the name Z stack of tubulin. We'll open this image and we will create maximum intensity projection, and then again we will crop this image at a desired area. Then we will set scale and we'll put scale bar. So final image will be look like this. The first image will look like this and the second result will look like this. So to open this Z stack of tubulin, I'll go to File, Open, and here you will find this Z stack of tubulin. Okay, so this one is hyperstack image with different XY channel, Z and time. Okay, so first what we need to do, We need to adjust the minimum and maximum gray pixel for display in brightness and contrast of this image. To adjust brightness and contrast, you need to go to image, adjust brightness and contrast. Before going to this, we'll put this scroll bar to seventh image so that you can see these structures slightly, and here we'll adjust its brightness and contrast. Okay, then next step is maximum intensity projection. For this, you need to go to image, stacks, and Z project. Here we'll take start slice 1, stop slice 14. And here we need to click this maximum intensity. And then, okay. So here we can again adjust its brightness and contrast. Okay. So result one is it looks like this. Next, you need to save this. We'll go to File, Save As, and .png with the name Max Z Stack of Tubulin. Okay. Next step is you need to convert this MIP to a green color image. For this, what you need to do, you need to go to LUT and select green. Okay, and next you need to crop this image in 30 into 30 micrometer square area from the center of the image. For this, we'll select this tool and we'll draw this square area in middle of the image. So here you can check its size in width and height. Okay, so this one is of 30 into 30 micrometer. Next, we'll crop this. To crop this image, we need to go to image and crop. Here you can see it has been cropped. Then next you need to add a scale bar of 5 micrometers. For this, first you need to do set scale. This one is set scale. For this you need to go to analyze, set scale, and you need to provide these values. So distance, okay, here we'll give this value 0.065 microns, and here this one is in microns, and 0.065 pixel is equals to 1 micron. Okay, then I'll click OK. And then again you need to go to analyze, tools, scale bar. Okay, so set scale is wrong, I think. We'll go to again set scale, and here Distance in pixel will be 1. Known distance will be 0.065. Aspect ratio 1. Unit of length micron. And then I'll click OK. Again, analyze, tools, scale bar. So here we'll put 5 micrometer of scale bar. Okay. So this image is similar to this result too. Next, we'll save this image. For this, we'll go again to File, Save As, .png, to billing, cropped. Okay, so this image has been saved. Here I'll wait 1 to 2 minutes so that you can complete all these steps. Meanwhile, I'll close this wall. Okay, now come to the next exercise. So, in this exercise, what you need to do, will open this image from the sample image folder, and as you have learned several filters, so from that we'll use Gaussian blur filter, and by using this image calculation methods, we will reduce the background of this image. I'll show this image first. So I'll open this. To open this, I'll go to open, and from this file I'll open this image. Here you can see this image has this background noise, and you need to remove this background. Okay, so for this, first you'll go to process, filters, and Gaussian blur. And here you need to use sigma radius of 8, and then press OK. You can see in preview that this image has been blurred. Now I'll click OK, and then next we need to save this image in a .tiff file. Save as tiff with this name, virus F1 background. Okay, next step is we'll use image calculator. For this, we need to open this image again. We need to go to File, Open, and open this image. And then next we'll use image calculator. For this, you need to go to Process and Image Calculator. We'll select here, we'll subtract virus F1 background image, from the virus F1. And then here I'll click OK. So to visualize this image, we need to go to edges, brightness and contrast, and we need to, okay, I think I subtracted wrong. I'll go again, process, image calculator. I'll select this F1, subtract. Okay, so here we'll go to image, adjust brightness and contrast, and we'll increase this. Okay, so you can see this one is similar to result 2. So in this way, if you have any image and it has some background noises, by using these steps and this Gaussian blur filter, um, anyone is having any question? Okay, so you can use these steps to clear the background noises. Is there any question, Vaishnavi, from your side? If you have, you can write in chat box. Vaishnavi, can you hear me? You can unmute yourself and ask. Okay, fine. Okay, so next we'll go to batch processing. So here you will learn something new, and we'll use one method which you have learned in previous lectures. So in this image, in this exercise, what we will do, we have a series of images. So if you have a different series of images and you have large images and you need to do some analysis, like if you need to put scale bar and you need to crop and you want to change colors of all those series of images. For this, you can use this image macros, and then you can do batch processing. So here I'll show that if you have this series of images, and if you want to crop in a particular area or size and you want to change its LUTs, or you want to put a scale bar in all these images at a time. So for this type of analysis, you can use this batch processing. So let's start this. For this, first you need to go to File to open this image. So here we'll go to Peroxisome. We'll open this image, Peroxisome 00, and then we'll open this. Okay. After this, next step you need to do is go to Plugins, Macros, and Record. So previously you have learned how you can record these steps and you can implement for further analysis. So here we will select this image, and first we'll set scale of this image. For this we need to go to analyze, set scale, and we need to provide this inputs. So distance in pixel will be 15, known distance will be 1, pixel aspect ratio 1, and unit will be in micron. Okay, then click OK. So here you can see that this step is recorded in your macros recorder. Next, you need to add a scale bar. For this, you need to go to Analyze, Tools, and Scale bar. So here we'll add scale bar of 5 micrometer. Then click OK. So here this step has also recorded. Next step is we need to crop an area of 300 into 300 pixels. For this, we'll select this tool and we'll crop this from here to here. To crop this image, we'll go to image and crop. So here this image has been cropped, and next we need to give different LUT. For this, we'll go to LUT, we'll give magenta color. Okay, so here all these steps have been recorded in your macros recorder. And next step is go to process. So from here we'll start batch processing. For this we need to go to process, batch, and macros. So we'll clear all these previous steps, and we just need to copy all these steps and we'll paste in this batch process window. Okay, and next we'll select this input folder. So here we'll select this proxy zone folder which has that series of images. And for output, you need to create one folder with the name batch processing. I have created it already. I have created it. So I'll just select from here output folder this batch processing. Okay, I'll wait 1 minute so that you can create that folder. Okay, so first step is we need to process this, but before going to this, uh, what we will do, we'll test one image whether this code is working fine or not. For this, we just need to click this test. Okay, so here you can see that it has cropped your image. It has given this magenta color, and the scale bar of 5 micrometer is already there. So if it is fine, we'll go to process. Here you can see that this software is doing its job. Okay, so now I think it's done. We'll go to this batch processing folder, and here you can see that all these images have been processed. These images. Okay, so by using these methods and this image macros, if you have a series of images of different experiments, you can use this method so that you can automate your process and you can do large analysis. So meanwhile, I'll close this. Okay, so next one is counting foci or puncta. So if you have any image, I'll show here. Okay, so if you have any image like this, if you have these types of foci or puncta or nuclear puncta, and you want to count all those puncta and you want to analyze or quantify the area or perimeter of those puncta, so by following these methods, you can do. So for doing this, first what you need to do, you need to this image HeLa cells from this image software only. And here I'll split channels. For this, I need to go to image, color, split channels. So mainly we will be working on this image which is having this different foci or puncta. So first what you need to do, you need to convert this image into gray. For this you need to go to LUT and grays. Okay, next you need to duplicate it into 2. For this I'll click here, duplicate it into 1, and I'll duplicate it again into 2. Okay, so now we have two duplicate images of this C1 HeLa cells. Next, so first we'll decrease the noise of this image. Here you can see there are several background noises, and since we are going to count these small foci, so while doing this job, this background may interfere. So before going to quantitative analysis, we need to remove these backgrounds. We are going to use this Gaussian blur filters and image calculator. That's why I duplicated this image into 2. Okay, so for doing this filtering, I'll just select this image 2, then I'll go to process, filters, and Gaussian blur. and here I'll give sigma value 2. Okay, then I'll go to 1. Again, I'll go to process, filters, Gaussian blur, and I'll give sigma value 1. And then next, what I need to do, I need to subtract image 2 from the image 1. For this, I'll go to process, image calculator, and subtract 1. I'll select subtract here and 2, 2, then click OK. So here, this one is subtracted image. Next, I'll change its brightness and contrast. So this one is your DOG image, and to change its brightness and contrast, I'll go to adjust brightness and contrast, and I'll increase this maximum value. Okay, so next, what we need to do, we need to do thresholding. So if you see in this original image before the removal of background, so you can see how thresholding is working with this. I'll go to image, adjust, and threshold. So here you can see this these foci are very you can see these are in very blurred quality, so we cannot use this type of image for quantitative analysis. So now we'll use this filtered image. So to transfer those thresholding to this image, I'll just select this and I'll click auto. So here you can see that all those thresholding has been transferred to this, and here we'll just change so that all those foci can covered. Okay, so this one is fine. Okay, then I'll apply this. So next step is we need to quantify these foci, but before going to this, we need to set measurements, like what you need area, perimeter, and what parameters you need to quantify. So for this, we need to go to analyze, set measurements, and from here we'll take area, mean gray value, standard deviations, perimeter, and here we need to check this limit to threshold. Okay. Okay, I'll just click OK. And then next, to quantify all these foci, you need to go to analyze particles, and here you need to give these values 0 to infinity, and circularity 0.00 to 1, and here we will select count mask, and you need to give display results, exclude on edges, clear results, add to manager, and the most important parameter is composite ROIs. Then you need to click OK. So here you can see the software has calculated area perimeter, width, height, and it has provided circularity. So all these values are from this filtered image. And if you want to transfer this this area which have obtained from the removal of background and after thresholding, if you want to transfer these value into the original image, for this you need to select this original image and I'll clear this. I'll select this original image and then I'll go to this ROI manager. From here you can transfer this. Okay, so you can label this for size, or you can select it like this only. And if you want to obtain these values from your original image, what you need to do, here you can save this, or I'll close this for now because I'm going to obtain all these values from this original image. So here I have closed this, and to obtain these values you need to click this measure from the ROI manager, and here we have all these values from the original image. Okay, so you can see we have obtained this result, all these values and values on your ROI manager. And if you want to quantify from a particular cell, for this what you need to do, first you need to go to this image. Okay, and I'll draw one ROI by using this freehand tool here. Okay, so before doing this, I'll close all these values because this one is new analysis from the same image of a particular cell. So if you want to analyze all these foci from this cell, what you need to do, you need to go to set measurements, and here you need to check this. So this should be selected importantly, and I'll click OK, and next I'll go to analyze particles. I'll give this values and I'll select OK. So here you can see, so here these quantitative values are from this particular cell only. And if you want to see this particular values of a particular foci, you can select from here, here, and here you can see all those values are moving, and you can select just measure, so it will add at the last of this image, this table. So here you can see. So I'll wait 1 to 2 minutes. Meanwhile, I'll close all these windows. Okay, let's come to the new analysis. So, in this analysis, what you are going to learn, if you have image of different cells and they are in different morphology, like some are in elongated and some are in circular and some are just going to divide, so that dividing cell will be having different morphology. And if you want to count a particular morphology of different cells, so how you can count this, uh, you need to follow these steps and this method. So before going to this method, you need to open one image from this images software only. For this, you need to go to File, open Samples, and from here, you will find this image with the name Blobs. So just open this image. Okay. So here you can see there are several cells which are somewhat elongated, some are small and circular, and some are just going to divide, or they are just divided. So to quantify, to analyze the morphology of these images, for what you need to do, you need to select this multi-point option. From here, this window will pop up. I'll just make it bigger, and from here you can select different type of this options. So here I'll select dot and I'll give this magenta color, and I'll select this XXL or extra. I'll select extra large. You can select others while doing your analysis. So first, I'll for first types of cells, I'll select this zero. This zero will denote the first type of your cell. So what you need to do, you just need to go here, and from here we'll select all those cells which are in bigger size. Okay. Okay, from here I'll count those bigger size images, objects. Next, we'll go for all those objects which are smaller in size. Okay, so smaller size of cells are 18 in number. Next, I'll go to third type of cells with the this two number, and we'll count those cells which have divided. Okay, so by using this method, you can count how many cells are there of a particular type. So the first one was bigger, 11 bigger cells are there, and then the smaller ones, 18 cells are there, and then cells which have just divided, they are 8 in number. And here you can screenshot this image, because if you are going to save this, these values will be not there if you are going to open that image again. Okay, so next move to this quantitative analysis. Okay, so if you have this type of different cells, and if you want to quantify the area of all these cells, um, smaller and larger cells, and if you want to um, analyze which cells are circular in nature and which cells are elongated. So by doing this quantitative analysis, at the end you will be able to quantitatively differentiate which are in circular and which are elongated. So for this, I'll open this image again. For now, I'll close this. Okay, so here I have opened this, and for quantitative analysis, first I'll go to analyze, set measurement, and from here you need to select all those parameters which you required. So here I'll check area and this fit ellipse perimeter, shape description, and okay. Okay, so before going to analyze it quantitatively, we need to do thresholding. For this, what you need to do, you need to go to image, adjust, and threshold. So here you can see, um, okay, you can adjust this value with the shape of your objects. Then I apply it. Okay, so in this image, what we need to do next, we need to invert this image. For this, you need to go to edit and invert. So here your objects are in white color and the background is black. So next, we need to analyze all these objects or your cells. For this, you need to go to analyze particles, and in this analyze particles window, you need to put these values. The size in pixel should be 10 to infinity, and circularity 0 to 1. And here we'll select outlines, display results. You need to select exclude on edges, clear results, and most important parameter, composite ROIs. then okay. So here you can see that these are area of all these cells, different kind of cells with this circularity and roundness values. And if you want to quantify this from your original image, from this, you can go to this, and here you can close this without saying, or you can save for your comparison. And here you need to select measure of this particular image, and if you want to do it for all, show all, and then select measure. So here we have these values from your original image. And if you want to differentiate in between the elongated and circular morphology of cells, you need to save this result morphology. Here we'll save this, and then next I'll go to that folder. I'll open that Excel file. So here we are having all those values, and what we want to do here is we want to differentiate all those cells based on its circularity roundness. So for this, what we need to do, we need to do some custom sorting. From here, we'll select custom sort and we'll add these levels. So here, based on this circularity and roundness and perimeter, we'll differentiate these cells. So here, we'll put smallest to largest value for all these levels, and then I'll click OK. So here you can see we have obtained all these values, area, and we have values based on circularity and roundness. So from here you can differentiate if your cells are having values towards the zero, they will be elongated, and those cells which are having values close to 1, they will be circular. So by using this method, you can differentiate your elongated and circular cells. Okay. So before going to next exercise or next learning, I'll close all these files. Okay, so next is mitochondrial network and branch analysis. So in previous lecture, you have been told that if you want to do some particular analysis, you can find several plugins and you can install those plugins, and you can use these for your a particular type of analysis. So here we are going to analyze mitochondrial network and the branch analysis. So for this we have provided one folder in this drive. From here you can download this and you can install with your images software. So I'll tell how you can install this. You need to download this folder from here and you can paste in a particular folder. So to install that, what you need to do, you need to go to plugins and go to install, and from there you can select that folder, and then in next step you need to install this in your Fiji folder. So in this folder you need to go to plugin, and here you can select from this image for the installation of that folder. So there are several plugins. So there are several plugins which can work with your window and this software, but most of the time or sometimes these plugins does not work. So in this case, what you need to do, just paste that folder in your image Fiji package, Fiji folder, and from there you can select those macros and you can use for your analysis. So if you are able to install this plugin, so in this plugin, you will find this mitochondria analyzer options here. And if you open this, you will find this window. And from this different options like 2D threshold, 2D analysis, 3D analysis, and 3D threshold options, you can do all those analysis. But in case if you are unable to install this plugin, what you can do, you as I said, you just paste this folder into your Fiji package and you can use this from there direct. So how you can use this, I'll show here. So to analyze this mitochondrial branches and network from this images software, you need to open this HeLa HeLa cell image. Okay, and then you need to split channel so that we can get only mitochondrial images. So for this you need to go to image, color, and split channel. So here I'll close all this. I'll put just mitochondrial image. Okay, so we have this image, and then we'll go to this Fiji app, and from here we'll open this plugin folder and mitochondrial analyzer and mitochondria analyzer, and then macros. Here you are having all these options which are present in those plugin. So from here you can use these codes directly. So next we'll see how you can use this. Okay, so from here we'll start analyzing these mitochondrial branches. So first we need to change its LUT. For this we need to go to LUT and change into in grays. and then we will convert it into 8-bit because these plugins and these macros will work with 8-bit image only. So to convert this into 8-bit, we'll go to type, image type, and 8-bit. And we need to change its scale from micrometer into microns. For this, what we need to do, we need to go to analyze, set scale, and here you can see it is in micrometer. So here we'll change into micron. And again, you can see this image has several background noises. So we need to remove this background. For this, we'll use Gaussian plus blur filters. Before going to this, we'll duplicate it into 2. Okay, so we have these two duplicate images. Next, we need to subtract the background from 1 to 2. For this, we need to go to process, filters, and Gaussian blur. So here we'll give this radius value to this second image 2. And then we'll go to this image 1, and then we'll go to process, filters, Gaussian blur, and 1. Okay. Then we need to do image calculations. For this, we need to go to process, image calculator, and we will subtract this image from 1 to 2. Image 1 is 1. Then we'll select this subtract, image 2, and then we'll click OK. There is something wrong, I think. Okay, again I'll go to process, image calculator. Okay, it has subtracted, but we need to adjust its brightness and contrast. Okay, here you can see. So now we have this image without this background noise. So this one is your DOG image. Okay, again we'll go to original image so that we can compare that how nicely the background has been reduced. So next you need to do thresholding of this image. For this you need to go to image, adjust, and threshold. Here you can see your original image is not that much good for quantitative analysis. So we'll do this from this image. For this, we need to activate this image. We need to do to transfer this thresholding to this image. And then from this image, we'll do this quantitative analysis. Okay, I'll apply this here. Okay. So next we'll do 2D analysis, branch analysis of this mitochondrial filaments. So as I said, you need to go to this macros, and from here we'll select this 2D analysis. We'll open this here by dragging this folder. Okay, here we have all those steps which can analyze this image. So simply we'll just run it from here. Next, this window will come. From here, you need to give parameters as per your requirement. So you can see there are several options. Wait a minute. You have this several options. If you want to count your mitochondrial filaments and you want to quantitatively analyze total area, mean area, total perimeters. And next, if you want to analyze the description of your mitochondrial branches, total branch length, mean branch length. So from here you can give all these parameters. And if you want to analyze area and perimeter at particular mitochondrial branch basis, then you can select this option also. And from here we'll just click OK. So here you can see. So from here, here you can see the count of total mitochondrial filaments, total area, total perimeter, mean aspect ratio, mean form factors, branches, branch mito per mito, total branch length. All those values are here. Okay, so apart from this, if you want to do another analysis, this display 2D mito ROI. So we'll just open this. So this analysis is for per mitochondrial. This macros is for per mitochondrial analysis. Okay, so we have this macros. We'll just select this. Before that, we'll close previous values. So here you will just need to activate this image and then just run this. So from here you can see this analysis is for particular mitochondrial branches. So by selecting this on ROI, you can see here all these options are changing. So if you want to analyze manually, or if you want to analyze a particular filament based on where is fission and where is fusion of these mitochondrial filaments. So by selecting this or this, you just need to, okay. So here you can select particular filament and you can just go to click measure. So here this, okay, this is the area of that particular mitochondrial filament. From selecting different mitochondrial filaments, you can just add those values by clicking this. This one is here. So you can go to analyze and set measurements, and based on those parameters, you can uh, quantify these values. So this one is for mitochondrial branch analysis and mitochondrial filaments analysis. So this one is so that's all for this practice session. And if you have any question, you can ask me now or you can ask later. And if you have any particular image and if you want to do analysis of those images and if you are getting any problem, you can send an email to us and provide the description of your image, what you need to do analysis and what you want. So based on your requirement, we'll try to um, create methods, combination of methods, and then we'll get back to you. Okay, so that's all for today. Thank you.